My name is Randy SCHEKMAN, and I'm the chair of the WLA Prize Committee for Biomedical Science. And the 2024 awardee is Professor Jeremy Nathans of the Johns Hopkins University School of Medicine. He is being recognized for having discovered the genes, regulation, and plasticity underlying human color vision and elucidating disease mechanisms that lead to blindness. Our story begins 300 years ago with the discovery by Sir Isaac Newton that sunlight refracted through a prism produces a series of colors from red through yellow, green, blue, and violet, with each angle of refraction corresponding to color of a distinct wavelength. 1 century later, the English physician Thomas Young hypothesized that the eye must possess distinct receptors for at least the 3 primary colors, and even before then, in 1794, the inherited variation in color vision, which affects 3 to 10% of males, was first described as color blindness by John Dalton. It was not until the 1960s that color receptors were localized to the cone cells of the retina. Cone receptors initiate vision through the capture of light using a protein called opsin that combines with a light-absorbing derivative of vitamin A to form rhodopsin. Jeremy Nathans discovered the molecular basis of color vision. As a new graduate student in 1983 at Stanford Medical School, Nathans single-handedly cloned the cow and then the human opsin genes responsible for color vision. This work revealed the first sequences of any sensory receptor. In remarkable work that followed in the 1980s, Nathans elucidated the molecular basis of color blindness. He showed that the tandem chromosome structure of the red and green photoreceptor genes lead to aberrant recombination, causing gene deletion. In his own lab, Nathans probed the molecular basis of tricolor vision. He mapped the amino acids in the protein sequences of the opsins that are responsible for the specific absorption spectrum of each protein. His lab followed this with the discovery of an elegant control mechanism by which a chromosome region exclusively activates either the green or red gene in a given cone cell. When Nathans began studying patients with inherited retinal degenerations, virtually nothing was known about the molecular basis of any form of retinal disease. Independent work from the laboratories of Nathans and Thaddeus Drija identified rhodopsin gene mutations as the first known cause of retinitis pigmentosa. In collaboration with Jim Lupski, Nathans identified the gene for Stargardt disease, the most common form of early onset macular degeneration. In an astounding tour de force mouse genetic and behavioral study, Nathans and collaborators demonstrated that genetically engineered mice, which are normally able to see only 2 primary colors, can become trichromatic when the gene for a long wavelength opsin is introduced into a mouse chromosome. This surprising finding highlights the remarkable plasticity of our visual system, in this case with the demonstration that the mouse brain adapted to interpret 3 colors. Nathans' wide-ranging curiosity, encyclopedic knowledge of the field of vision science and its historical roots, and the depth of his thinking and the creativity of his approaches places him among the best neuroscientists in the world.